first of all, I would like to congratulate Muriel and the team on a great cost initiative, and let's hope that things go really well in the future. And thank you also to Emmanuel for this invitation. And I just said to her, I had fun preparing this, so I hope you have as much fun watching me telling you about it as I had preparing it. Actually, I was looking this morning on the internet for a series of cartoons related to water on Mars. When I was at the Johnson Space Center, I remember seeing these series of cartoons of Martians on Mars under different hypotheses of water on Mars. We had Schiaparelli with the canals, and then the canali, and the Martians were sort of in their boats wandering around on these canals. And then the next stage we have the Mercury missions, which showed that there was no water on Mars. So these poor Martians were in the desert. Where's the water? Where's the water? And then further stage, and we can see with the orbital images on the surface of Mars that there's evidence, geomorphological evidence of water on Mars, and then we get the mineralogical indications that there was water on Mars. So the Martians are walking around with umbrellas because there's water falling down, there's rain, so they've got rain. And then they have they're at the edge of the beach, edge of the ocean, with their feet in the water. So and then previously, we'd still thought that Mars, even though there was water on the planet, that it was rather dry still. And just recently, last December, we had some more evidence of the fact that there was quite a substantial amount of water on the planet at some time and at some stage. Anyway, sorry, that's a sort of I couldn't find the cartoon series. I'll find it someday. And that's the introduction. But what I want to talk about are basically biosignatures for rocky planets. And for rocky planets, I'm talking about not a planet like Earth today, but the early Earth. A planet like early Earth, early Mars. So, why? Well, these are the types of environments that are probably far more common in the universe than the Earth today. The types of organisms that live on these planets will be far more common than the aerobic, the complex life forms that we know on the life today. So that's the rationale, and I'll go a little bit into more detail why and wherefore. What I want to do is to relate the biosignatures to habitability. This workshop, the general theme is habitability, but biosignatures form part of the context, which is habitability. You don't get biosignatures on a planet that's not habitable. So I thought it'd be interesting to go back to the global average conditions of the early Earth and early Mars. And taking, for example, rocks about 3.5 billion years old on the Earth, what can we learn about the environmental conditions from them, and making suppositions about early Mars, and really suppositions. So temperatures on the early Earth, we think, were probably relatively high, especially at the rock-water interface. And the rock-water interface is where everything's happening. This is where we're getting the prebiotic molecule complexification to whatever simple life forms arrive. There's high heat flow from the early planets, so there's a lot of heat coming out. A lot of heat means also a lot of volcanism, a lot of hydrothermal activity. So basically, hot local conditions, whatever the global conditions were, because there's still some discussion about the temperature on Mars. Anyway, so apart from the temperature, pHs of the waters probably slightly acidic because the atmospheres consisted mostly of carbon dioxide. A tiny, tiny bit of oxygen produced by abiotic processes, photolysis of water vapor in the upper atmosphere, radiolysis of water at the surface of any water bodies, and so on. Therefore, relatively high flux of UV radiation to the surface of the planets, 54 watts per meter squared compared to 1 watt per meter squared today. And if you make it DNA weighted, it goes up to 1,000 watts in the worst case scenario. So I spoke about the volcanism and the hydrothermal activity. Also, because the planets were still, there were lots of bits and pieces left over from planet formation, still quite a high rate of impact activity. So what we're looking at are planets where the conditions are extreme naturally. And this is where I like to say that these planets like these, are the norm. Our present-day Earth is anormal, and that we are the extremophiles, and not what we consider extremophilic bacteria. But that's my personal view. Now, I've spoken about the habitability on the global scale, but we need to be looking at habitability from the microbial life scale, the first cells, primitive cells, simple cells. And these scales, spatial scales, time scales, are quite different to those that we find on the whole of the planet. Well, there have been various estimates. How long does it take for life to appear? We don't know. Some people will say tens of thousands of years, some people say millions of years. It's debatable, but in any case, we need bodies of water that contain the ingredients of life, the carbon molecules, the nutrients, energy sources, etc., that are stable over these periods of time scales. So we're looking at bodies of water of at least 100 kilometers in diameter, if not more. However, this is different for established life, because once life has become established, if it can get to a newly habitable environment, for instance here, this is an effluent from a hot spring in Yellowstone, and the grey is the silica sinter, and the green are photosynthetic microorganisms. But it's just to show, that where an environment becomes habitable, microbes, if they are in the vicinity and can use the nutrients that are available there, they will colonize this environment very rapidly. And the environment might last only for a very short time or for a longer period of time. So timescales for established life are variable, but much shorter than those for the origin of life. In terms of survival, well, we don't know exactly how long microbes can survive in the long term. I mean, they've been described from salt crystals, from amber, etc. So that could be on sometimes millions of years. Now, there's also the effect of habitability and time, geological time scales, because habitability changes as the planet evolves. On the Earth, for instance, we started off with a basically carbon dioxide atmosphere, but with the appearance of photosynthesizers and then oxygenic photosynthesizers with oxygen pumped into the environment, the habitability changed. The planet became habitable for another kind of microorganism, more complex organic forms. But on Mars, this didn't happen. On Mars, Mars, because of its small size, it cooled down more rapidly, it lost its protective magnetic shielding and lost most of its volatiles, and so Mars basically became a desert planet after since the Hesperian, let's say, we don't know exactly when, but let's say between 3.8 and 3.5 billion years ago. So whereas it was habitable early on, today at the surface, it's inhospitable at the surface, maybe habitable subsurface somewhere, but it's certainly inhospitable at the surface. So what I'm going to talk about, the biosignatures, goes on a sequence of premises, at least with respect to Mars. Habitability on the surface was heterogeneous in scale and time. We think that life could have arisen because we think Mars had the ingredients for life, but it could have arisen at different times and at different places on the planet. Conditions for continuous evolution didn't really exist. Whereas Earth was basically an ocean planet, early Earth was an ocean planet, Mars has always been a landlocked planet. We're not too sure about the longevity of the Northern Ocean. That's a factor to take into account. With the rapid degradation of surface conditions, there was limited possibility for the evolution of life. And so this is why I'm concentrating this introduction on small primitive life forms such as anaerobic chemotrophs that are thought to appear before the phototrophs. What kinds of organisms could these be? Here are some examples of modern organisms. For instance, the modern archaea, Pyrococcus yanashi, is anaerobic, hypothermophilic microorganism. It's a chemoorganotroph. That means it uses organic matter both as a carbon source and a nutrient source. Or chemolithotrophs, such as this thiothrix. They obtain their carbon from carbon dioxide in the seawater and their energy from inorganic sources. And why not viruses? They're organisms as well. Why not something like viruses? So what I'm going to... biosignatures are the signatures of these life forms, whether they are extant, in which case, in terms of Mars, we're looking at subsurface organisms, possibly, or extinct, in which case these will be the organisms or the traces of organisms that we might be able to find on the surface of Mars. And I've put here some photographs of fossil structures that might have been related to these kinds of archaea, similar shapes, similar sizes, filaments are similar to the chemolithotrophs, and these are fossilized viruses down here. I would never be able to identify these, by the way. Anyway, so I'm going to now concentrate on the signatures of extinct life. Generally, biosignatures refer to a whole range of the characteristics of life, the actual physical structure. It could be a cell, colonies of cells, biofilms, or even physical three-dimensional structures that these organisms create, like microbial maps on the surface of sediments that interact with the environment, such as the MIS, which is microbial induced sedimentary structures, or even stromatolites. And we'll hear a little bit, I think, about microbial constructions. But then there's also the organic components. Life is formed of water and organic molecules. The organic molecules have certain molecular compositions. They have elemental compositions. They have also particular structures, chirality, and so on. And how do we test for, what kind of signatures of microbial metabolism could be preserved in a rock record as a fossil? Well, microbial metabolisms produce biominerals, minerals that are precipitated or formed in association with the biological structures, either directly or indirectly. Microbes also concentrate in their organisms elements. They can leach elements out of rocks, the nutrients that they need. The fractionation of certain elements such as carbon, sulfur, iron, etc., is also influenced by microbial metabolic activity. And then even more in a more ephemeral way, microbes can have an influence on mineralogical features, such as they can dissolve the surfaces of them, or the presence of organic matter can influence the size and the shape. So there are a whole range of possible biosignatures. Now, I'm a geologist, so I have to, none of this really makes sense unless you put it into a geological context. So the geological context gives us information about the local habitat, the physico-chemical conditions, because if the feature that you're interested in, if you want to know, is it really related to life or not, you need to understand, you need to understand the geological context. You need to see the relationship of the biosignatures with respect to their direct environment. So this is absolutely fundamental to any study of biosignatures. Now, I was speaking about biosignatures in general. if we're looking for just anaerobic microbial signatures, what do we find or what don't we find? We find everything except these three-dimensional structures and the microbial mats that are formed by oxygenic, or not necessarily oxygenic, photosynthetic microorganisms. So all of these can still be potential biosignatures. So we're not looking for stromatolites, we're not looking for these microbial mat influenced structures. Now, how do these biosignatures preserve in the rock record? I'm just using a series of cartoons to give you an overview. Well, imagine you have your sediments, and these particles are your sediment particles. They're coated with colonies of chemotrophic organisms. They degrade, they're buried by more sediment, they degrade, and the organic matter just gets mixed in with the sediment. So this is one form of preservation where you have the sediment, but just a sort of a goo, a matrix of organic molecules associated with the particles and sometimes directly fixed to the particles. Some of these molecules can be very important indicators of life. They can be related for instance, carotene can be related back to pigments and so on, and some of them can be related directly to microbial features. Hopanes can be related to cell membrane features. However, these so-called biomarkers don't last eternally. They get degraded. What happens is that the organic molecules, the functional groups, are gradually removed during diagenesis, during geological time. They become degraded, and then by the time we get to early Earth, 3.5 or Mars, 4 billion years ago, we are left with aromatic molecules, polycyclic aromatics, which we find also in carbonaceous meteorites. They're also abartic molecules. So, this is one form of preservation. And then another form of preservation is when the actual microbial structure, if it's particularly robust, becomes sedimented with the sediments, and it just gets squashed in the sediments. And in fact, Emmanuel Javot's been doing some nice work on these squashed microorganisms, if I may disrespectfully talk about them like this. The remains are there, but they're just squashed. The feature is there, but it's flattened. And then a third way of preserving these organisms is, or these biosignatures, is by precipitating, or when minerals precipitate on the surface of the structure, mineral-coated organisms. And this happens actually very rapidly, very easily. I've done a lot of experiments, killing millions of microbes in the process, I'm afraid. But the minerals in solution fixed to the cell walls. We have here a thin section of a microorganism. The scale bar is 0.1 micron. And all the black points are minerals that start fixing on the surface. And they polymerize and form a kind of crust. The organic matter is trapped inside and can be in anaerobic conditions. It can remain trapped. Otherwise, in oxygenic conditions, it will be oxidized. So the organic matter can be preserved, but you end up with a crust. And this is an example of one of my studies from rocks from the early Earth, a filament, microbial filament that's been silicified. The rock is almost 3.5 billion years old, but there's still organic carbon associated with the feature. So these features can be preserved. However, the actual preservation starts with diagenesis. That's the alteration of the original organic matter and the precipitation of minerals, or etc. Whoops, sorry. But then the features have to be preserved through geological time. All sorts of things happen. There can be earthquakes, erosion, the rocks can be buried, they can be heated up by metamorphism, impacts, whatever. So there's a lot of serendipity to the preservation of the biosignatures. Now, there are two other points that are equally important. One is you might find a structure in a rock which you think came from a microbe, but are you sure? Is it something abiotic? And then another aspect is, okay, you can decide that your feature is, yes, it did belong to a microorganism, but did it form at the same time as the rocks, or did it come in later? Syngenicity. Just to give you some examples here, I'm showing in the your upper left some silicified structures that I've interpreted as silicified microorganisms in a 3.5 billion year old rock. And I'm comparing them to abiotic silica spheres. Superficially, there's a lot of similarity between the two features. And so a lot of work needs to be done to really make the distinction between abiotic structures and biogenic structures. And in terms of syngenicity, were the structures formed, if they are biogenic, were they formed at the same time as the rocks? We have to do quite a lot of work, very high detailed work. In fact, all of these images that I'm showing you, we're using high resolution scanning electron microscopy. Keep this in mind, because when we get to Mars, we can't export a scanning electron microscope to Mars, at least not yet. So, but you know, there are all sorts of details like embedding in being embedded, coated with minerals, embedded in the matrix of the rock, which help to determine whether the feature was original or not. This is important because old rocks are old rocks. They're full of fractures, and microbes love these fractures, especially if there's original organic matter in them. And here these are photographs of surfaces of rocks 3.8 billion years old from Isua in Greenland. And on these surfaces, they're coated with endolithic microorganisms. These are microorganisms that actually go into fractures. They live in the rock in hot or cold deserts. And in this case, we're looking at fungal hyphae and microorganisms that themselves have been fossilized in situ. They're younger than 8,000 years old because the area was covered by ice until 8,000 years ago. But this complicates the situation. Anyway, getting back to early Mars and early Earth. So that's my sort of general introduction. So the geological context, I want to speak a little bit about that because I am also a geologist. So what we're looking at then on early Mars, and we'll see also on early Earth, are basically basins of water, impact basins filled with water, in which sediments are deposited. These are volcanic sediments. The early planets were volcanic. Forget about your nice sandy quartz beaches or your shelly beaches. Didn't exist. We're looking at black beaches, volcanic sand beaches. And they form sedimentary structures, big dunes-shaped structures that we can interpret, we can identify the environment of deposition of these sediments, shallow water sediments. Same thing on the early Earth. Again, I like the excuse to do some sketches, although I'm not a brilliant artist. But this is, imagine this is the environment on the early Earth. It would be similar for Mars. You have your volcano spewing out ash, shallow water environment with dunes forming underwater. The sediments are being deposited on basalt rock, lava. The whole environment is influenced by faults, a lot of tectonic activity, also perhaps caused by impacts, and then a lot of hydrothermal seepage, hydrothermal fluids coming up through various vents and faults. Plus, we have to keep in mind we've still got all the extraterrestrial organic matter and meteorites, etc., coming in. So you've got a mixture of things, of life processes going on down here, with all the meteorites and the carbonaceous chondrites and etc. coming in, and high UV. I'm going to show you an example from a beach area. This is, but before I do that, I'm going to show you the modern equivalent. Forget about the green, etc., and the odd cow or whatever in the field. Just imagine this is the tidal environment, where in the bay of Mont St. Michel in the northern, in France. But look at the ripples and the tidal channels here. You can see the nice ripple marks here, cut through it, and you can see all this sort of cross-bedding. This is the vertical section of these ripples. Now, we see exactly that on the early Earth. Thank you. Sorry, dears, just hang in there. Another 5 minutes. Another 5 minutes. Anyway, so what I'm trying to say is that on the early Earth, we have exactly the same situation. Volcanic sediments, we can see that we can get the geological context from cross-bedding, etc. And in these rocks, I mean, these rocks are formed by volcanic grains. These are the volcanic grains, which we can see with optical microscopy and transmitted light. Now, I'm going to show you a series of slides using Raman microscopy, and there are quite a few talks in the biosignature session using Raman microscopy. Keep in mind that Raman microscopes are going to be sent to Mars, so this is why Raman is important. But this is one of the volcanic particles, and in blue we can pick out, it's coated with carbon. It's coated in carbon. The Raman signature tells us that the carbon is old, it's mature, it's consistent with the metamorphic grade and the age of the rocks. And again here, another example, that one was from Australia, this is an example from South Africa, and here the carbon is in green, coating volcanic particles. So we have quite a lot of carbon there, but again we have to use higher powered techniques to determine what is this carbon, what's it made of. And we can see that there are sort of carpets or monolayers of these balls that are full of carbon. These balls actually turn out to be fossilized microbes. You can see beautiful microbial features such as the cell division here, and this individual's deflated lice, so we've got life and death. I'm not going to go into the details of these because this is not the aim of this talk. The aim is just to give you an overview. But these are the kinds of details that we need to determine biogenicity. Can we do this on Mars? Again, some of these, in fact, I'll skip these ones. I want to go on to Mars now. Getting to Mars and the search for signs of life on Mars. We have Curiosity on Mars at the moment. ExoMars 2018 should be leaving in 2018, and Mars 2020, which is the first in the step of missions for return of samples from Mars. Now, Curiosity, what instruments does Curiosity have that could search for traces of life? Well, there is a microscope, Marley, with a resolution of about 36 microns. The individual cellular structures that I've been showing you from the early Earth are less than 1 micron. So Marley can't actually see biosignatures. There are organics, there's a laboratory with organics that can analyze organics and isotopes. And in fact, this is a picture of Marley, and this is an image of the kinds of things that the microscope can see. But the laboratory was able to detect recently the presence of methane. Now, methane could be a biosignature, a gaseous biosignature. Also, on the ExoMars mission, there are various instruments, an infrared spectrometer, or various infrared spectrometers, Raman spectrometer. They can identify organics. But a laboratory, an LD GCMS in the laboratory that can also analyze for organics and isotopic signatures. On Mars 2020, there is a Raman microscope, BASTA. So, what could we find in situ on Mars? Frankly, anything that needs optical analysis, we can't do it. We can look for carbon, the presence of carbon, and if the material is analyzable, it's not too refractory or something, we can look for molecular composition and fractionation of carbon, and perhaps chirality with the ExoMars instrument. So, however, these are the only things that we can do in situ. But of course, if we bring samples back in the terrestrial laboratory, we can look at all of these features, keeping in mind we still need the geological context, so we still need the in situ study in order to determine is what we're looking at biogenic or not, and we need this information also to help us to. We don't just want to say, okay, we've got life on Mars, we have a cell or something. We want to say more. We want to say, what was it? What was it doing? Why was it there? What's the relationship with the environment? Yes, of course, the first person who finds traces of life on Mars might get a Nobel Prize, but it's more than that we want. So okay, there's some really great talks this afternoon and on Wednesday on biosignatures, so thank you very much.